Hello there and welcome to another edition of the Evening Review. My name is Okito Greg, and in studio with me this evening we have Professor Lamech Mwapagha. He is the Associate Professor for Medical Biochemistry at the Namibian University for Science and Technology, NUST. Good evening, Professor.
Thank you very much.
You are here to tell us about the conference, Cancer Genomics Conference,
Yes.
which will take place from 24th to the 26th of September at the Hilton Hotel.
Yes.
What is cancer genomics?
Thank you very much for having me on your studio. Cancer genomics is basically a field that looks at the whole genetic makeup of an individual.
Okay.
So you're looking at this whole field of the genetic makeup of somebody, the DNAs, and then you're able to determine whether this person has cancer or not.
Okay.
Basically in a layman's term. Right. So you're able to identify those bases called A, T, C, and Gs, and you're able to see whether there's a mutation in any of them. Using, of course, techniques such as sequencing, whole genome sequencing, and high throughput techniques.
I see.
Yes. Yeah.
Okay. And what is the use of determining if somebody has cancer or not?
Or the use of using those techniques to determine whether somebody has cancer or not is that you're able to come up with good treatments, what is known as personalized medicine or precision medicine.
Okay.
A good example would be, let's say, if 2 people or 5 people are sick, having a headache, then they go to the hospital, then they're all given Panadol.
Yes.
Right? That's the normal, let's say, treatment.
Yes.
So you'll find in those 4, 1 or 3 might get completely cured of the headache, 1 might have slight side effects, and the other one won't even get cured or even might have adverse effects.
Yes.
But with this, what is known as precision medicine or personalized medicine is whereby you will now end up sequencing each individual.
Okay.
Then you're able to tailor the medicine based on the individual. So that's what is known as precision or personalized medicine. So you're tailoring the treatment to an individual, which is far much better.
Yes.
Yes. Yeah.
But I imagine it would be a lot more costly.
Yes, it is. It is quite costly, especially when it started this work on whole genome sequencing. When they first sequenced the human genome, this was in the early 1990s or 2000s. There, the US are the ones who started doing it, and other companies. So it used to be quite expensive to do that, and it would take a whole lot of time, close to a couple of days or even weeks, for them, months even, for them to come up with the human genome sequence. But nowadays we have got equipments which are known as next generation sequencing equipments. There are various companies which have these equipments. So you are able to have your genome sequenced in 2 days, sometimes even in 24 hours. The problem comes in when it comes to analyzing this data. Remember at the beginning I said you have got those bases A, T, C, and Gs. So when you sequence somebody's whole genome, you end up with 3 billion bases of this. So this is a lot. So imagine you have got these A, T, C, Gs, 3 billion of them. So you need now bioinformaticians to make sense of it by loading it onto databases. So it used to be quite expensive, but nowadays the prices have gone down to around $1,000 per sequence. Yes. Okay. And they're still going down.
They're going down.
Yes. So that's the reason why we have this conference.
I see.
Yes.
Let's expand a little bit about the conference and also about your role as chair of Conference Scientific Committee.
Yes. So the conference, the idea, as I just mentioned here, the conference starts on the 24th to 26th. But before that, on the 23rd, we have a pre-conference, which is taking place at NUST, Namibian University of Science and Technology. So what we are doing is that from the morning session, we'll have trainings for early career scientists on bioinformatics. As I've just said, this is the techniques for reading those letters. So this is a critical skill that Namibia really needs. We don't have that critical skill of bioinformatics. So we are going to be training people on bioinformatics, the early session. So we have somebody coming in from Cape Town who is a bioinformatician. And then in the afternoon, we'll have proteomics training. So this is also somebody from CSIR who is very good at proteomics. So these early career scientists will be trained on these 2 aspects of bioinformatics and proteomics.
Well, what is proteomics?
Proteomics is whereby you're able to study the proteins, how they are arranged and how they network between each other.
Yes.
Right. So all this is tied up together. There's what is known as omics technology. So you have proteomics, transcriptomics, genomics, and also metabolomics. So all this is tied together. And then in the evening of the 23rd, we have a Vice Chancellor's public lecture being given by one prominent professor who was this year named Times 100 Most Influential Person in the world.
Okay.
Among the 100 is called Professor Christian Happy. So he'll be giving a talk in the evening, a public lecture, which is, we'll be releasing the poster very soon, in fact today if possible, or latest tomorrow. And then now the conference starts on the 24th now at Hilton. This pre-conference is at NUST.
Yeah.
Then at Hilton now we have 3 days. The first day we'll be talking about what it entails to do cancer genomics, and we have various speakers who are coming to discuss on this. Then we'll also have sessions on AI, and because the AI is now the big thing.
Yes.
So AI and cancer screening, because you'll find that if you go to hospitals here, that we have a hard time when it comes to screening of cancer. Most of the time, like in prostate, they have to go in and take a biopsy.
Yes.
But if we can be able to use this by just screening with the AI, let's say looking at the eyes, and you're able to screen, and you're able to determine when somebody has cancer or not, that's a game changer.
Yes.
And then the second day, the third day now, and that's the 26th. So we'll now have a session where we'll be talking about the future of genomics. We have 3 panel discussions. So all these discussions will be revolving around how best are we going to utilize cancer genomics as Africa, and how best can we come up with a database that is for African database, right? Because Africa has the greatest genetic diversity,
Yes.
right? But we still haven't utilized that genetic diversity. So you'll find that the database that we use, even like during that period of COVID, we are getting drugs from outside where they are using sequences made from Caucasian. But now if you have a database for Africans, it's easy to come up with medication or drugs that are tailored towards our own genetic mechanism. So those are some of the discussions that we'll be talking about. Discussions also around how do you protect this database? How do you protect genetic information of individuals? One interesting fact about Namibia is that we have a community here called the Khoisans. It's known already that in their genetic makeup, they have 25% genetic makeup, which is not found anywhere else in the world. So this is a big deal for Namibia. So bringing this conference here in Namibia, it will bring these conversations and how best can we learn from those people who are already using cancer genomics, and us here, how we're able to interact and learn about the whole process of genomics. So my role basically is to be able to bring in these speakers from abroad, people already using cancer genomics, and then link them with also our participants here, plus our clinicians who are here and academicians who are in Namibia, and to see how best whether there can be a synergy. I'm sure there will be, and how best we can come up with a way forward when it comes to cancer genomics.
Wow, it's very interesting.
It is
Just to take us back to an issue that you mentioned, the application of AI.
Yes.
And I know that in the past, when AI applications were tested and rolled out, it was often found that there was bias which was negative or detrimental to our African communities, even not recognizing African people in terms of facial recognition and other AI applications.
Yes.
Has the technology progressed beyond that, or is that still one of the barriers that you hope to overcome with this development?
So what I can say about AI is a model which is trained.
Yes.
Yes. So you'll find that initially when AI started coming about, or the people who generated AI at the very beginning would be, let's say if it's in your house, you will start in-house, first of all, before. So if I come and use something that's in your house, won't really be modeled to me, but modeled to you. So that's how AI was. So you're actually right that initially we had these challenges, but right now AI is being trained. It's being trained a lot on different aspects. Apart from the usual things of, let's say, being able to identify different cars and all that, but it's also now being trained when it comes to diagnosis. We know that you can actually use AI to look at X-rays and be able to tell where the problem is. So in my research group right now, I'm collaborating with an organization known as Oncotech, who are also part of the funders, part of the sponsors of this conference, and who are also coming. The COO is coming here. So what we are using with this is we are able to use a machine or just a phone. So that's got an app which is known as an OptoScan app. So you're able to look at the retina of the eye and then be able to tell different types of cancers from there, and even type 2 diabetes.
Wow.
So this has already been done before in other countries. So what we are trying to do is we are trying to validate this AI here in Namibia.
Yeah.
So if we're able to validate it here and it's able to work, then it's going to be rolled out across Africa.
Wow.
So we stand at a very, so it's quite interesting.
Groundbreaking.
I can
But
say.
I'm interested in your reference to Namibia, and I'd like to know, how far has cancer treatment in Namibia progressed? What's the gap between the reality on the ground in Namibia that a cancer patient might face and the groundbreaking work that you are doing now? When do you think we would get to a point where we could apply these scientific breakthroughs for our own people?
For our own people? Well, that's a very good question, because it's quite pertinent what you're talking about. So the idea for this conference is to be able to translate what we are doing. So initially, what people used to do is just do, so there was a disconnect between the clinicians and the researchers.
Yes.
So the researchers would be doing their own thing, getting results, publishing, that's it. The clinicians would be doing their own thing, using drugs, everybody was separate. But what is happening now is that we have realized that it's impossible to work in silos.
Yes.
There has to be a synergy. There has to be a harmony between the two and all the other stakeholders involved. So to answer your question is that we are very close to achieving what you're saying in terms of precision medicine. In that, right now we are in my research group, we are doing research which involves looking at gene expression. How are genes being expressed in patients? So we are doing, in my group, we have researchers in different type of cancer. One is on head and neck cancers, one is on prostate cancer, the other one is on liver cancer. So we are looking at what genes are being expressed. And we are working closely with the Windhoek Central Hospital, with the clinicians who are there.
That's very good.
Yeah. So the results we are going to get, other than publishing these results, but before we publish, the agreement is that we have to go back and present and say, look, this is what we found, and this is what we think is happening in our patients. What should we do? Because there are also databases where you can actually get a sequence, blast it, which gives you an idea of what treatments to use.
Yeah.
So that's what we are trying to do.
Excellent.
So within a short time, we should be able to translate what we are doing in the labs.
Speaking of time, Professor, we have to take a quick break,
Okay.
because we've got an ad that's going to run now for a minute, but we'll be right back.
Thank you.
Thank you.
Thanks.
Welcome back to the Evening Review. My name is Ogito Greg, and with me in studio this evening, I have Professor Lamech Mwapagha. He is Associate Professor of Medical Biochemistry at NUST, and he's telling us about the upcoming Cancer Genomics Conference, which is about to take place later this month from the 24th to the 26th of September. Professor, let's expand a little bit about the conference itself. How many participants are you expecting here in Windhoek?
We're expecting around 150 participants.
Wow.
For this conference.
And will they be coming mostly from our neighboring countries or from further afield?
From farther afield. We have people coming all the way. There's one arriving tomorrow from Burkina Faso.
Oh, wow.
And then we have some coming all the way from UK, from US, from China even. So we have a number of people from across board. Yes.
And this work that you are discussing, it's quite groundbreaking in terms of the science and the medical application of it. Is it applied already elsewhere in the world?
Yes, it is actually applied. Yeah, we have a good example, I would say, is a young man called Nick Volker some years back, who had a stomach ailment. So what they did, they couldn't be able to find a cure for it. So what they did a whole genome sequencing, and they were able to identify what the problem was. And then they were able to treat him through this whole process of genomics.
Very good.
Yes, so it's something that is already being applied. That's just one example.
Yes.
There are a number of examples of whereby cases of sickle cell anemia, there are a number of examples. Yeah.
You mentioned earlier that the results from the conference will first be shared with the clinicians here in Windhoek at the hospitals.
Yes.
I wanted to find out in general, what is the situation in terms of support for such groundbreaking scientific research and application here in Namibia? Do you find that there's an adequate framework? Are discoveries protected in terms of intellectual property? Is the environment conducive to developing these breakthroughs further and bringing them to practical use?
Yeah, I could say there is support. The beauty of of Namibia is that we are sort of like starting on a clean slate,
Okay.
right? Because most of this work has not been done. So it's easier for us to look at what other people have done and then don't repeat the same mistakes.
Yes.
So that's the beauty of us doing research now in Namibia. So we are at a huge advantage. So we will not be wasting a lot of time, a lot of money, a lot of reagents and all that, because we'll be able to see what others have already done. And then we'll now be able to see what we shouldn't do.
Yes.
And then move forward. So on the issue of IPs, before we used to have what is known as helicopter research.
Okay.
Where helicopter research is whereby scientists would come, do research here, and then go back, or come take our samples, then go back and do the research, and then no credit is given to the researchers here.
Yes.
So that's what is known as helicopter research. But nowadays that one is no longer there. So what we are able to do, we currently now have principal investigators who are actually here on the ground. And that's what even Africa is advocating for.
Yes.
That if you want to do research with people here in Namibia or in Africa, then the person who is responsible, principal investigator, should be from the home country. And then you are coming in to support.
Yes.
Right. So and then we also now we sort of like have strong IPO systems. Like I know at NUST, we have a huge office that is responsible for IP, an organization known as DRIP. Right. So it's responsible for research and also for IP. So if you have got anything to do with research before you even start, you engage them and they're able to advise on how best to go about it and how to protect you when it comes to IP or any patents that you come about. So there are measures. We still have a bit of long way to go, but there are processes that are already in place.
And progress being
progress. Progress being made, yes.
Excellent, excellent.
Yes.
I'd like us to go back a little bit and touch on the human aspect, the people involved. I know I lost my mommy to cancer, and there are many Namibians who suffer from this scourge, either themselves or family members or people they love. Is this research a source of hope for the future?
Yes, it is. Indeed, this is a source of hope. Because as you have said, all of us, in one way or another, we have been affected in one way or another. Either a close relative like yourself, your mother, or a friend, or a neighbor, or somebody. So all of us in one way or another have been affected when it comes to cancer. So this gives us hope.
Yes.
And that's the reason why I decided to have this meeting here in Namibia, whereby we can engage.
Yes.
We can engage and see, okay, this is what is happening. There's a high cancer cases rate now going across board, across Africa, not only in Namibia, across Africa. Why is that the case? Because this used to be a disease which people would say is for the whites or Caucasian or for the rich.
Yes.
But nowadays you find even the poor person is getting cancer.
Yeah.
So what could be the case? Is it that we have changed our diet, we have changed our lifestyle, or what is happening? So this conference is going to get us talking, right? What could be happening on the ground, and how best can we move forward from this, right? How can we leverage on what we have currently in our institutions, in our government hospitals, and what are some of the policies that can be put in place to support cancer care and treatment, especially for our patients? Because cancer genomics, research in genomics is also a bit expensive.
Yes.
So how are we going to discuss this model also of pricing and costing? And how can we be able to come up with funding models that are able to support the treatment and care of our patients? So those are some of the discussions so there is future and there's a hope.
Excellent.
That's what I can say.
Excellent.
Yes.
As Associate Professor of Medical Biochemistry at NUST,
Yes.
I'm sure you're also aware of some of the other research directions that are being pursued, some of the other things that are happening within that department or field. Can you share some of that with us?
Yes, there are a number of research going on in our department. We have, other than my group, we have a group that looks at what is known as the Moringa plant. It's led by Professor Kwamba. So he's able to use this plant to be able to find different ways even of cleaning water. You know, we have a big problem when it comes to clean water especially. So he has been able to show that you can use these seeds for that. And now we are looking at how can we use these seeds or finding the active components when it comes to treatment of cancer. We also have other groups that are looking at biodiversity. So this is led by a marine biologist, Professor Moregi, looking at the different biological or ecological aspects in the country. So he's working with kelp blue in the Lüderitz and the rest. We also have other groups which are looking at food security by Professor Pasi Chimamurombe, where he's looking at maramabin. So there are a number of research. And in my group, other than what I've spoken about in terms of cancer genomics, we're also looking at what is known as microbiome.
Okay.
So microbiome is basically, humans have got a lot of bacteria in them.
Yes.
So these bacteria outnumber human cells 100 to 1.
Okay.
They outnumber human genes 10 to 1. Okay. So that means there are a lot. That
Yes.
means you are working bacteria. So research has shown before that children born via cesarean section are more prone to allergic conditions like
Yes.
asthma and the rest. But those born via the normal route are less prone to these conditions. Meaning that shows you that as they pass through the normal vaginal route, they pick up the bacteria, which kind of protects them. Whereas those who are born by cesarean don't pass through the normal route.
Yes.
Right? So they don't get sensitized to those bacteria. So you find that nowadays there's a good researcher known as Rob Knight. So what he did is when the wife gave birth by cesarean, he took a gauze and then wiped the mother's vaginal canal, birth canal, and applied the gauze on the kid's mouth, nose, and ears.
Yes.
So in a way to sensitize the kid.
Yes.
So this is a whole lot of research that is going on in that. And one of my master's students even published a paper recently who is trying to look at the role of microbiome in head and neck cancers.
Okay.
So they could be playing a role.
Yes.
Yeah. So there's a whole lot of good research currently ongoing.
Excellent.
Yes.
I'd like you just to revisit for a few moments the pre-conference that you were speaking about.
Yes.
And especially about access to young people who would like to pursue studies and their own personal development in these scientific fields, how they can participate, and generally what they should do to prepare themselves to pursue this kind of research.
Uh-huh. So one of the things that, maybe to answer the first one, is on what is the pre-conference about?
Yes.
Right. So as I said at the very beginning, from 8 to on the 23rd, this will be taking place at the HTTPS, Lower Campus NUST,
Yes.
High Tech Transfer Plaza Select building.
Yes.
So there, what we'll be doing from morning session 8 to 12 is a training on bioinformatics. So looking at how are you able to look at these bases and how are you able to delineate or to be able to understand what is happening in the genetics of somebody, like the genome
Yes.
of somebody. So that's the morning session. Then the afternoon session is proteomics,
Yes.
looking at how these proteins interact with each other. So for you as an early scientist, career scientist, to get into this field, first of all, there has to be an interest. There has to be a passion to it. You have to have an interest in biology, an interest in cancer. So if you have that interest and have that passion to want to learn more, you want to get to understand more on what is happening in our everyday lives, basically that should be something that should push you forward to come to that. And the only thing is that for those trainings, because it's specialized trainings, we are only restricted to 40 participants.
Oh, okay.
Yeah.
Yeah.
So, but currently we do have, I think maybe 1 or 2 spaces remaining,
Okay.
but it's restricted to only 40.
Yeah.
Because it's specialized where you require the person who is training to also interact and engage with the participants.
Yes.
So even 40 is a huge number.
Yes, yes,
But
He agreed to go. They agreed to go for those numbers.
Okay.
Yes.
I'd like us to go back to the beginning again.
Yes.
And just for the benefit of our wider audience.
Yes.
What is cancer? And how does it develop so that you can look at genomics and try and trace it?
So in short and long, cancer is just the abnormal growth of cells, of normal cells.
Okay.
So your body system is supposed to be working perfectly the way humans were created.
Yes.
It's supposed to be working perfectly. So it has what is known as an immune system.
Yes.
So this immune system just runs around checking your system every time, right? Anything wrong, it's eliminated.
Okay.
Dead cells, once they're like red blood cells, they get to 120 days, they expire, they're removed from the system. So that's how your body works. So what is known as senescence, as you age, as the cells keep on aging, then of course they are removed from the system.
Okay.
Right. So now, when these systems are now not working optimally, so you have got genes that are known as tumor suppressor.
Okay.
So the name itself tells you that they suppress tumors.
Yes.
Or any gene that is not working well, it's supposed to be removed, or any cell.
Okay.
So what could happen is that in your everyday life, depending on your lifestyle, you could end up picking up certain things as you go along.
Yeah.
Let's say excess of alcohol, excess of smoking, your lifestyle. Let's say you pick up a virus, HPV or HIV. This, what it does, it integrates into your genome.
Yeah.
So then it ends up changing the sequences. So your cells now start growing abnormally because the genes that are supposed to protect are now mutated, are damaged.
See. Yeah.
So now your cells start growing, and they start growing at a much faster rate, so what is known as cell proliferation. So as they keep on growing at this faster rate, then they form what is known as a tumor. So once they form a tumor now, let's say, just for example's sake, let's say this is a breast cancer.
Yeah.
So, and then it's not discovered early at stage 1, it's discovered late, maybe at stage 4, stage 3, or stage 4.
Yes.
So what will happen is that as the cells keep on growing, they do what is known as invasive. They keep on going inside and inside. Then finally, they enter the bloodstream. One cell or a few cells.
Yeah.
Once they enter the bloodstream, they are carried, and then they go and lodge to another place.
I see.
So that process is known, they spread. That process is known as metastasis.
Okay.
So when they go to that other place, they form another secondary tumor.
I see.
So you had breast cancer, you end up having liver cancer.
Yes,
So when you're treating breast cancer, let's say with radiation,
Yeah.
if you found it late, they could have spread, what is known as metastasis.
Yeah.
So that's why you think somebody went to remission, but then it comes back.
Yeah. Professor,
Yes.
our time is finished.
Oh.
So unfortunately, we have to leave it there. But thank you very much. It was very interesting and very informative
Thank you.
and empowering.
Thank you.
I wish you all the best for your conference.
Thank you very much.
Thank you very much.
Thank you. Thank you.
And that's all for the evening review this evening. But join us again tomorrow evening when we'll be discussing a different topic. Thank you very much. Good night.